Carrie did all of her degrees at University of California at Davis. She played on the women's basketball team, varsity women's basketball team, and I think she had to give it up her last year because engineering was too...
Last 2 years.
Last 2 years. So she played on the women's varsity basketball team as a second-year student at UC Davis as an undergraduate, so she was a really good basketball player.
I would say it's JV. I don't even remember.
Okay, all right.
I was confident.
So she went on to do her master's. She did her undergrad in chemical engineering and then her master's and her PhD in civil engineering. She and I had the same 2 advisors as graduate students, but I'm older than Carrie, so I graduated 7 or 8 years before she did. I will say, since she's joined our faculty in, I think, 1996, after I saw Carrie speak at a conference in Denver, and I was wowed by her presentation for 2 reasons. Number 1, she gave a great presentation, but number 2, she was filling in for somebody, and she wasn't even involved with the project. So she stood up and gave this great presentation on somebody else's work at a conference. And I came back to UT, and we were looking for a young air pollution person. I said, I have just the person, right? So we ended up interviewing Carrie. She showed up, and she's been a champion for our undergraduate students the entire 20 years she's been at UT. So Carrie, I think, is going to talk about a project called UT Biome today a little bit. UT Biome is a project where she's turned the University of Texas into a living laboratory, and she's had over 600 students, mostly undergraduate, who go around the University of Texas campus to Waller Creek, to the football stadium, to insides of buildings, and they take swab samples, they collect water samples, and they do biological analysis. So she's doing this detailed microbiological analysis of the entire University of Texas campus with students. So it's a tremendous educational program for students. And she also started about 15 years ago something called GLUE. GLUE stands for Graduates Linked to Undergraduates in Engineering. She started this program, and since she started the program, there's been 650 undergraduate students who've been paired with graduate students to do research. So we talked a lot about wanting undergraduate students to get research experiences at the university. Carrie's somebody who's taken the bull by the horns and said, I'm going to make it happen. And she's making it happen with these 2 wonderful programs. Over 1,200 undergraduate students involved in these 2 programs. So great asset for our department, great asset for our university, and I'll leave it to Carrie now. Thank you.
I thought this was a great picture to start with because the 2 folks that are represented there on the left is Wiley Jennings. He just graduated from our program with a master's degree. And several of the projects I'm going to be talking about, the research area I'm going to be focusing on today, involved him heavily. And that means he spent weeks in the lab doing DNA extractions, which is a very labor-intensive yet meticulous process. And I lost the other. Well, and the person on the right that you can no longer see is...
Sorry about that.
Oh, that was you controlling it. I thought I had some control here. Was J.P. Maestra. He's a research engineer in our program. And, for instance, mapping the UT biome couldn't have come off without his involvement. He would march out to the creek. 100 students would visit him over the course of a day, and he'd have everything laid out, all the logistics, and the students had a fabulous experience. So I want to acknowledge all the folks, and that's only part of the group that contributed to the work I'm talking about today, but those 2 were involved very heavily. So what I'd like to talk about today is, well, it's entitled Inside the Space We Build, the Indoor Microbiome. You probably can't recognize that picture because I purposely modified it. This is actually the inside of the Natural Sciences Library at UT Austin. But I like this artistic effect where it gives it kind of a speckled look. So that, yes, there are people sitting here and there, but as far as a microorganism is concerned, you're really just a surface to colonize. Okay? And so I'm going to talk about this relationship between microbiome, or the indoor microbiome, built spaces, and why we care about them and why engineers should get involved in this area. So the first thing, I actually borrowed a slide from Dr. Corsi because he does a lot of work in the indoor area. The first reason is we spend all of our time indoors. At least in the United States, we're spending nearly 90% of our time indoors. And what that looks like, of course, varies. So on the top, you'll see a residential home in Austin, a commercial building, and the bottom one there, I was fascinated by Hong Kong. Obviously, the built environment is very different there. But as Americans, we spend nearly 90% of our time indoors, so our exposure to many contaminants and certainly to microorganisms is going to be dominated by what's also present in that environment with us. And we're not alone. So you think about, obviously, your family, if you're thinking about your home, maybe your pet, but in reality, there are microorganisms all around you in the built environment. On the left there, you see a surface that is going to come in contact with your hand every time you open the door. Well, because your hand, the skin microbiome that's associated with your hand is going to be transferred to that door handle. Anybody want to guess what the upper right is? I think you can. Showerhead. Showerhead? How many people looked inside their showerhead? How many people are going to go home and check their showerhead now? Exactly. So, and that has built up over time in a showerhead. It's not atypical, actually. And the lower right here is your home heating, ventilation, and air conditioning filter, your HVAC filter. You see the dust that builds up on that. Well, we've actually been exploring what else is in that dust. And what can it tell us about the microorganisms that are present in these indoor environments? And in fact, we have plenty of company. So you may have remembered from your high school biology days where you'd get a swab and they'd send you home to take a swab of a surface and come in and you'd put it on a plate. That's the culture-based world that we lived in for a very long time. And you look at the colors and you're like, ooh, that's interesting. Then we started to do something, the technology came along, and we started to do what we call high-throughput sequencing of the DNA that we can recover from different types of samples, dust samples, air samples, water samples. And the world got a lot more interesting. So each one of these circles, I think like the smallest, the limit of detection for just what we chose for this particular slide is almost 9,000 what we call reads, DNA sequences. So they had to have a minimum of 9,000 to be associated. Each one of these colors is a different species. Okay? And when folks did side-by-side comparisons here, if you're just looking at the bacterial community, they're estimating that less than 2% of what was actually present, say, on a surface or in the dust was actually platable. You could actually pull up on a plate. The majority of it was hidden from view until relatively recently, and just like we're looking at the human microbiome and using these same techniques, we've now started to look at the buildings that we occupy and see what influence do we have on those buildings, both in how we design them, but also in how we operate them, and frankly, just our mere presence in those buildings, what impact does that have? So we now realize there's an incredible diversity of microorganisms present in our indoor environment, and we're starting to investigate, well, are these important or not? And the question comes about, well, why do we care? Okay. The bad news is we care because there are certain diseases, and childhood asthma is just an example of that, that the rates of childhood asthma in the United States are rapidly increasing. I think it's on the order of like 9 to 10% of children have had asthma or been diagnosed with asthma in their lifetime. It's the leading cause of childhood disability in the United States. And there are many different triggers for asthma. Typical ones, allergens, but you've heard of mold, otherwise known as fungi, bacteria. Even the presence of moisture indoors can be a trigger for, asthma trigger for some folks. And there's certainly others. So I call that the kind of the bad news. Why are we seeing this rise in the prevalence of childhood asthma? Is it associated with the fact that we spend most of our time indoors? There's a real active area of research that's addressing that question. But I do have a little bit of good news, and you're wondering why you're looking at cows. And that's because it turns out we're not only looking at, okay, some of these microbial communities, microorganisms, may be potentially detrimental to human health. There's actually now a whole line of research looking at what we call the protective microbiome. And believe it or not, which you may believe, but there's a farm-like microbiome. There's some researchers in Finland who we're collaborating with. And it's also been confirmed as well in the United States that microorganisms associated, and actually cow barns is where they're specifically been looking at them, are protective against the development of asthma if the child is exposed to this microbiome during the first year of their life. So we often talk about the bad news, which is, okay, there's microorganisms all around, and we should always be worried, or we should always try to control them. But there's a whole line of research now looking at what I call the positive microbiome that is actually protective for human health. Moving on, there's obviously what you hear about are potential pathogens in the indoor environment, and this is a huge issue, especially if you're talking about hospitals or any place where you have a vulnerable population. So there's many to choose from, but a few stand out. Legionella. So, for instance, in the recent, what I would call disaster in Flint, Michigan, you hear a lot about the lead contamination. What you don't hear much about was the Legionella outbreak. And I think the number is up to 12 people now that died from Legionnaire's disease as a result. So Legionella is a huge concern. You might have heard about it from the cooling tower problems that they had in New York City. They also had a Legionnaire's outbreak. One you might not have heard about, and I'm going to talk a little bit about today, is what we call NTM, non-tuberculous mycobacteria. This is naturally present in tap water, municipal water, soils, but it also is responsible for an incredible number of pneumonia cases, serious pneumonia cases in the United States. So these are waterborne, okay? You've also heard about the development of antibiotic-resistant strains. MRSA being an excellent example of that, but there's grave concern about the development of these strains, and particularly when you're talking about healthcare-associated infections, there's a huge push to improve what we say are the numbers here, right? So if you look at the numbers, and they vary, okay, depending on what source and what they're counting as what we call HAIs, but well over 1 million infections, and the original estimates were up to 100,000 people die associated with these infections every year. If you look at the latest CDC numbers, they've been looking at the percent improvement from that original data in 2002, and the good news is there's been substantial improvement. The bad news is they haven't solved all HAIs, so certainly going into a hospital and acquiring a second problem is really not what you're interested in. So there's a lot of push here to try to control what's being done medically, but also the environment that these folks are in, since they're often immune compromised. How can we make sure that they have the best outcome possible? Then there's another good news associated with this, and this is very recent work, but I think you've probably read about it a little bit in the popular press about the positive microbiome. Probiotics is a term that you hear a lot about, right? There are there are species that are associated with protecting human health. In particular, gut microbiome, probiotics and gut microbiome. And I've just started working with this fabulous group, actually one of our other Air Force grads, and he's been working with some people at UC Boulder, looking at the links, for instance, between microbial exposures and mental health. And in fact, they found species that they believe are protective for positive mental health outcomes. So there's quite a bit of work going on with that. And this is very new, but it'd be nice to look at the microorganisms in the indoor environment, not just from a negative point of view, but perhaps we could look at it and say, what could we do to encourage the development of the healthy microbiome that protects human health? And that's really what I'd like to do, is redefine what we mean by healthy buildings. I don't just mean energy efficient, water efficient. I mean protective, microbially friendly, if we will. So where do these microorganisms come from? You see, you might have a hint right here. Look to your neighbor. Look to the other neighbor. Wipe your hands. Okay, us. Okay? We are walking, seeding bacterial machines. Okay? I think the estimate is like 100,000 to 200,000 skin flakes per minute are being shed. So by the mere act of walking into this room, sitting down, you are seeding this room, as has everybody before you, with your skin microbiome. Okay? So human-associated bacteria are found throughout homes, multi-use buildings, and hospitals. It's been worked by Rob Dunn and others at UC Berkeley looking at what's the human-associated bacteria in residential homes. So as you can imagine, if you were to swab a pillowcase, that might have a lot of human-associated bacteria present. However, the indoor door trim, how many people clean the indoor of their door trim? Excellent. You can come to my house. I'd never do it. But anyway, it's a passive site that's collecting kind of settled dust, but we often don't have direct contact with it. So it's going to have, it still has the human imprint, if you will, of our microbiome, but not nearly the same level that you would at the, for instance, the pillowcase. Also, the mere fact that you walked into the room, and if you're like me, I'm a little bit of a shuffler, right? What you're doing is there's dust on the floor, there's microorganisms associated with that particulate matter, and as you walk into the room, you kick it up, okay? So resuspension of the dust from the floor, and if you look at what's in the floor dust, you know, these are rough numbers in these particular studies. They said roughly 20% are human-associated microorganisms. So the good news is there's this kind of this normal background flora that's present, has always been present. So just because we just now are getting an insight to the diversity of what's present doesn't mean that we suddenly need to be like, you know, paranoid. But what we do need to understand is what's kind of the normal flora, what you mean by that, I mean what's okay, and what do we need to be worried about? And that's a really active area of research. And I do like my dog, so just, and Dr. Corsi really likes his dogs, if you know him. For those who have a dog, they found that the, Rob Dunn's group found that there's a very strong impact on the bacterial flora. So they are also seeding the indoor environment with their particular flora. Oh yeah, sorry, it's 200,000 to 600,000 flakes per minute. There it came up. Okay, so this is, where do they come from? But this isn't the only sources. Just to name a couple others, outside, certainly the microorganisms that are present in the soil and the air, they're certainly working their way in, okay? And in particular, if we're looking at airborne, fungi, the fungal community that you have indoors is heavily influenced by what the fungal community that's outdoor, in the absence of water or moisture contamination where you're flourishing, or you're growing, you know, a particular species on your, I don't know, ceiling tile or whatever. Water is a very important conduit for microorganisms into the home, and I'm going to talk a little bit about showers. They are the perfect bioaerosol generator, if you think of them. Not only do they serve a purpose, but they also have, your tap water has a natural background of microorganisms in it, and we've been starting to look at those. So not only the water that you bring into your home intentionally, but also the water that unintentionally gets onto your different building materials. So the top one there is some green building materials that was meant for cabinetry, which I think is particularly not a good idea given that it started to grow very easily. And then you see things like this inorganic ceiling tile that no matter what we did, we inoculated, that stuff was tough. They use it on indoor swimming pools. Huh? Inorganics and actually pH 10. Turns out it's high pH too. Exactly. So what built spaces are we studying? I'm going to give you a few examples. And I like my flying organism because it's so happy. So we've been looking at residential homes, particularly semi-rural homes is one of our bigger studies that we've been looking at. Also, we've been looking at showers in homes, and also the HVAC filters. So if you think about a filter, it's in place for a nice long period of time, extra long if you never change it, And so it provides an integrated sample of the airborne microbial community that's present in that environment. So we're doing what we call filter forensics. We go in and we say, well, what's in that filter and what can that tell us about the indoor environment? We're also doing a lot of work on classrooms and schools. So this is, again, an interdisciplinary projects. We work with the School of Nursing. We're working with the folks in architectural engineering, Attila Novoselak, Ing Zhu, that you were just mentioned before, and also Rich Corsi is working with us on both of these classroom and schools projects. Also, we've done a series of studies looking at retail environments, so these large, generally large retail stores. And I don't have time to talk about that, but if anybody's interested, I can tell you the results of that study. And then I want to kind of end with talking about mapping the UT biome, which Rich mentioned earlier. It's really been a blast to actually take the students out of the classroom, go down to the creek. We do have a little lesson about poison oak and other things first, but and sample the creek and look at the water quality in the creek, but also we've not only looked at the outdoor environment, we're looking at the indoor environment. So we've sampled, like, for instance, the air within classrooms, and I'll give you just a quick example of one of the projects that the students have done. So let's start with homes, and in particular showers. What we did is, actually, there are the researchers. There's JP in the middle. These are two former master's students, Chloe and Alex. There you can hardly recognize them. Now they're not there to be protected from whatever's in the shower. They're there to protect our samples from their skin microbiome. So that's why they're in what we call the bunny suits. We went into a series of residential units, actually some in San Antonio, some in Austin, and took a bioaerosol sample of the air in the shower before the shower was turned on, during the shower operation, and after the shower operation. And this is just showing some of the results for Austin, Texas. It's a surface water source. Monochloramine is the chlorine disinfectant. Is the chlorine residual type. And we use this, we get millions of sequences out of these things. And we use a whole bioinformatics pipeline, CHIME, and other things, as well as this source tracker program to try to figure out, of the organisms we found in the air, where did they come from? Okay. So in this particular case, so we not only sampled the air, but we took swabs of the ceiling, the floor, the walls, etc., in each of these residential showers. So in this particular example, you can see that even before you turned on the shower, the air, the microorganisms in the air looked a lot like the biofilm growing on the ceiling. How many people clearing their ceiling? Not even you, not even door trim person cleans their ceiling. Okay, I got you. You are now. You're going to go home right now, right? No.
Yes, I am.
And during the shower, you can see that the floor microbiome was kicked up as the water hit the bottom of the shower. You can see that the water microbial community is now represented in the air that you're inhaling, and the ceiling swab remains. The good news is when you turn off the shower, you still have a little bit of the floor community, and the ceiling community is pretty dominant in that sample, and there's a little bit of the water community left. Okay? So it gives us an idea that if we're looking at what you're exposed to in a shower, is it of concern? Well, I'll show you in a moment. You can decide if it's of concern or not. But it also tells us where did it come from. Because just telling me, not to take a shower is really not a viable option. But giving me some sort of solution. Should I be looking at the water? Should I be looking at the shower head? Should I be looking at, what should I be looking at? Okay? And so we're interested in what are we being exposed to? Is any of it of concern at all? And what should I do about it? Because I really do enjoy my morning shower. So if you look at the two cities, we did Austin and San Antonio, and we took a lot of samples. We got excited, right? So we took air samples, and what ambient means is really like in the house, but not actually in the bathroom. The air sample before the shower was turned on, during, after. Plumbing biofilm samples, surface biofilms from the bathroom, and we did it both in Austin and San Antonio. And each one of these colors represents a different genus. Okay? If I put wrote down every single what we call operational taxonomic unit that you detected, it would be really a mess. Okay, these are really diverse samples. And without knowing much about DNA sequencing or anything else, if the color scheme looks roughly the same, it's roughly the same community on a very kind of gross measure of it. Okay, so you can see that these are pretty different communities depending on where you sample. But also what comes to mind is, remember the mycobacteria that I talked about? Well, what we found is that very little of it was present, I'm sorry you can't see it here, but it was present in the San Antonio samples. But it was pretty prevalent, these bright yellow bars, it was pretty prevalent in the Austin samples, okay? And so that tells us, and if you start to look at it, you see it's pretty, occasionally you see it in the air, okay? But you see it a lot in the biofilm samples. And it's known to like biofilms. So that tells you that, well, one direction you might want to go is, well, biofilms are important, and can we improve our premise plumbing system, our shower heads, in order to mitigate the development of what I would call unhealthy or potentially unhealthy biofilms. So that tells us kind of where we're kind of looking toward. Can we provide some sort of solution here? And again, for you and I, we've all been taking that shower for forever in our residential home. We're fine. Again, though, if you're immune compromised, that is where you have to really pay attention. So if we're talking about a hospital environment or somebody who's undergoing chemotherapy and has gone home, then you have to start really paying attention to what type of microbial exposures they're getting. And actually, Wiley had asked a bunch of his colleagues to take pictures of themselves with their filters, and Mitch, who came to us from Berkeley, other than the fact that he's wearing the California shirt, which I forgave him, I like the fact that he put on his helmet, he's a former football player, to be able to handle that filter that's so dangerous, I guess. So you see these different filters, we have different types of homes, and we were looking at the relationship between what we find on these filters, remember that filter forensic idea I mentioned earlier, and the, essentially whether there are children with asthma in that home or not. And so in our study design, we had 40 homes of children with asthma, and another 20 control homes in the same area there was no children with asthma in the home. And what we're trying to do is look at, is there any relationship between what we're finding on those filters and either the presence of asthma or even the severity of that asthma for the kids in those homes. And we did it both winter and summer, because you can imagine that we have what we call run times on your HVAC unit. Winter is darn hard to catch here. Summer is really not hard to capture because we have very long, hot summers. So this actually, what I'd shown you previously, is if we'd taken a look at the bacterial diversity that were present in these filter samples. So we did 60 homes twice. These are kind of the more dominant genera that were present, and basically the bigger the circle, the more there was in that, the more there was of that particular genus in these samples. And if you look at these kind of range here of organisms, the ones that I've just circled are actually associated with humans. Okay, so it shows that we're seeding our environment. Some of the ones that are the most dominant here are actually human associated. And there we are again. Where do the bacteria come from? We did some repeated samples. This was actually off of a single filter, so we did it many different times, taking many different subsamples and compositing them. Bottom line, if we can compare what we're getting from the bacteria from those filters from bacteria that grow on human skin. And you can see, for instance, that about 5%, there was an outlier here, but generally about 5% or so are associated with human skin bacteria. If you look at fungi, instead of a human and being there with the cloud, you'll see that we have a nice outdoor scene. So fungi really are heavily influenced by the fungi present outdoors. Okay, so they're really outdoor associated. and it'll vary from place to place. I had to go here. Sorry, you've all eaten your lunch. Who else is there? We are in Texas, and there are cockroaches in Texas. I hate to break it to you. What we decided to do, though, and this is actually some of the work that Wiley and JP had done, is that we'd looked for some cockroach allergen, done some testing, some ELISA testing, and it's really kind of painful to do. Then we found out that there's this Bladobacterium species that's an obligate symbiote in the cockroach gut. In other words, if there's a cockroach, it has to have this bacteria in its gut. And so we decided, well, let's go look at our filter samples and see what we find. Do we want to know? Oh, come on. Fun. Can't be engineers without a little fun. Okay, houses reporting cockroaches actually had this bacterial species present in the filters, which meant the filters were actually giving us a good capture of the bacterial community present. And so we looked at two different types of homes, detached homes and manufactured homes. And we looked at the relative abundance of this particular type of the bacterial species. And if the homeowner said, no, we really don't have any cockroaches, I've never seen one, we actually didn't detect it in the filter. If they said yes, we detected it using this, kind of surrogate measure. Now, this is just kind of interesting because we all know that there are cockroaches around Texas, but there are certainly different levels of cockroach infestation. So again, it's getting at this idea of what can we discover from using some of these filter forensic or sampling protocols? What can we learn about an indoor environment? All right, what about microbial diversity and asthma? Okay, so this is the first, I think, 20 or so homes. This is kind of the preliminary data set. We've actually got the all 60 homes, both seasons, and we're looking at this big metadata set now that we're doing the analysis. But some of the earlier analysis that we saw is that if you look at the relative diversity of the fungal communities present in the homes, and whether there was intermittent or persistent asthma for the kids in those homes, yes or no, you'll actually see a lower diversity than on average, than you see in the homes without that, asthma symptoms. Now, in this case, it's not statistically significant, but we're looking at the overall data set. But it does follow trends that have been reported by others, that actually in homes where kids do develop asthma, that tends to be lower fungal diversity. So this is one of Jordan Petchia and Karen Danmiller had done some of this work looking at samples from, I believe it was from California. If we look at bacterial diversity, you see the opposite trend. And again, we're trying to confirm this, so this is very preliminary. We're trying to confirm this with the broader data set. But if you look at the microbial diversity of the bacteria, it goes the opposite direction. Greater bacterial diversity was actually found in the homes with this intermittent asthma or persistent asthma symptoms versus those without. So we're going to see if this holds for the whole data set. But others are actually starting to see something similar in different data sets. So they're taking them in California and different parts of the country. So again, we're starting to get some clue. We don't know why. What's driving it? Why does lower diversity for fungi and higher diversity for bacteria, why would that have any relationship to asthma? We need to work on that. But it does at least give you some hints on something to address. All right, and now I'm going to switch over to a project that I've been working with actually the people in architectural engineering very closely with, and also the School of Nursing has been helping us as well. And I have, as I mentioned to a few of you, I have 2 kids in high school in Texas, and they have spent time in these buildings. So I was motivated, let's just put it that way. There are over 500,000 of these things in the United States. 500,000. So you put 20 to 30 kids in each one of these, and you do the math. It's a lot of kids spending a lot of time in these portable buildings. As Attila notes, they're portable, but they're certainly not temporary. Okay, they may be there a decade or longer. Okay, they go and they sit, and they're pretty much there. Previous studies by CARB, California Air Resources Board, and others, some folks at Berkeley had found that, just kind of a general walkthrough, they found poor ventilation, water intrusion, higher formaldehyde levels. And one of the things is that we were concerned concerned about, well, what's the microbial community in these systems, in these portables? And Ying Zhu was looking at the semi-volatile and volatile organic contaminants in these portable buildings. And so our objectives were to just look at the effect of how the ventilation systems were working in these systems. So there's Attila doing a blower door test. And one of the key things is to investigate the relationship between the microbiome in an occupied space, where everybody is, and what I call the hidden spaces. Ceiling plenum, crawl space, wall space, okay? And we don't want to just say, oh, there's problems, if there are, or there's no problems. But if there are problems, we want to identify some low-cost solutions to those problems. All right? Just telling people, well, you know, it's not the best, but I got nothing for you. That's not really what an engineer does. We want to try to figure out something that we can do. So we're still in the is there a problem phase, but I'll give you a few hints here. So this is just a artistic rendering, if you will, of a portable, where I won't say it is, but anyway, a portable classroom in Austin somewhere. And Attila found some pretty interesting things. We found that the fresh air vents, see that duct tape? Bad use of duct tape. Also, a lot of filter bypass, so they weren't necessarily in great condition, and it really depended on the age of the structure and how often it was being maintained. And I would say, you know, these schools are trying their best with this infrastructure, and we just want to say, you know, perhaps you should do this or that to improve ventilation. In particular, we wanted to look at, if this is just a kind of a simplified rendering of what you might find in one of these buildings. You've got a crawl space, an attic, you've got the outside. And really what we want to do is look at these hidden spaces, I call it, particularly the crawl space, the attic, and if there's a wall space, which actually turned out to be relatively rare in these things because they're all, you know, they're pretty tiny wall spaces. You know, what is the relationship? Do you pull in any of that material into the indoor space? Despite, you know, in addition to the ventilation, some of the ventilation issues we looked at. So the good news is, if you positively pressurize these buildings, which means you blow fresh outdoor air in, guess what? The indoor air starts to look like the outdoor air. So that's the good news. And what we did is we looked at the indoor air community in a building that was actually kind of near neutral pressure. So it would swing between positive and negative, okay? But if you look at the green here, the green there on the top there is the filter dust. So what we were detecting in the filter dust is what we were seeing in the indoor air. Again, there's that source tracker idea. The filter sample was pretty similar to what the indoor air sample was because it was doing a lot of recirculating of indoor air and not a lot of fresh air was coming in. Fresh air here is, which one's fresh air? Oh, outside air is red. Okay? So this is what we kind of estimated using this technique was the contribution of outside air to the microbial community in the indoor environment. And this is all bacteria. So we positively pressurized the space. I don't know, it's like 6 or 7 air changes or so. And then we resampled. And the good news is, the outside air, we successfully flushed with outside air. And you see more of a reflection then of the microbial community outside is now inside. So some of this is evidence for perhaps positively pressurizing a space. Also, if you look at another kind of interesting feature, though, is you see the blue there? We use the door handle as a surrogate for human contact. In other words, human skin biota, because everybody has to touch the door on the way in. If you use that as the human imprint, kind of a marker for that, you'll see despite the fact that we increase the outside air community, for instance, you still got your human, you can't just flush out all the human-associated bacteria. They're gonna stick, if you will, okay? But still, it does give you some idea of what would be the effect, for instance, of positively pressurizing some of these spaces. Okay, so then I wanted to just end with talking about this Mapping the UT Biome project, which has been a lot of fun, really. It started a couple years ago, actually. It hasn't been going that long. We got some funding to run a pilot scale project, which has now run its course, from the, it's called the LIFT program. It's some UT Austin funding. You bring IT, if you will, and other things into the classroom. And so the objective was to engage the UT Austin community in environmental data, and specifically we had targeted biological data, but it really morphed into looking at the environmental conditions, indoor air conditions, in different types of environments. So you all know about Waller Creek. I've been down there a lot, and I've suddenly, since the beginning of this project, I'd spent like 17 years on campus. Yeah, there's the creek. I'd never once gone down there. And now I'm obsessed with the creek. I watch it, I take pictures of it. Every class has to look at the same pictures of it. But it does suddenly make you very aware of the environment around you. And really what I wanted is the citizen, everybody calls it scientists, but my argument is citizen engineers are also good people to collect samples from across campus. How many people took 341? Back in the day, Introduction to Environmental Engineering, a few people. Did you ever leave the classroom? Yeah, we've changed that. Now every year, all the 341 students get up out of the classroom and go out and sample something. And we wanted to not just do these projects. You've heard about all these different projects, and then it ends up on somebody's computer and goes nowhere. So we developed an interactive mapping program. So I worked with Dr. Pasalakwa. She does a lot of hydraulic engineering, but she's also an expert in GIS and ArcGIS. And we basically took all the data, at least from the first year. We have multiple years that we haven't gotten up there, but first year's worth of data. We put it on an interactive map. So you see these dots. You could go up, particularly the ones near ECJ, you could click on it and it would give you the data that the students had collected that semester. So we have the first 9 months or so of the data. And then really what we wanted to do is get this started. And then you could imagine all sorts of future sustainability efforts that the students could be involved with. Right? You could, look at the CO2, or global warming impact of the energy use associated with different buildings and map it onto the campus. You could look at water usage. You could almost anything and use this kind of interactive mapping so students could add to the database. And then what I have now is that when, for instance, students get data from the creek, they also have to go back and look at the previous year's data that their colleagues collected and look at it over time. It's not just a single, here's my one grab sample. So it's nice because it starts to build this library, you will, of data that the students themselves have collected. We've had different sampling campaigns. This was, we actually weren't able to do it this fall, but we're hoping to start, this spring, we're hoping to start again next year. We did an indoor air quality classroom study. There was a building renovation, so we did the baseline sampling before the renovation. We did a laboratory building looking at the ventilation effects. We did the Waller Creek ×5. Everybody got to visit the creek. I became obsessed with it. It's a beautiful creek, really. We also looked at what I call water stagnation, premise plumbing. So I once had the students sampling the premise plumbing in ECJ, and I did not realize that all the deans were meeting on the 10th floor. So my students show up with their bottles, their things, their gloves, their glasses, and they're sitting there as all the deans and stuff are walking by. I'm like, ooh, should have checked the schedule. But anyway, we had a great time. So this is just showing some of the instrumentation, since we're talking about indoor microbiome, that we used. And again, what we do is we take, basically we rated his lab, don't tell him, we rated my other buddy's lab. We went and we got all the equipment that we needed for the students to do real-time measurements. And, for instance, this is an Impingor to look at bioaerosol sampling. This is a button sampler to do the same thing, but particle associated. And there are actually nearly 700 students now, I think, have been involved. And again, I have to give credit to JP because he's been the motivation behind this. So one of the studies, one of our first studies, we did what we call the outside to indoor gradient. We looked at sampling, well, a building you may or may not recognize. We did outside the building, we did in the entrance area, we did the hallway, and then the doorways right here, we did inside this classroom. Okay? And this happened to be an air pollution engineering course. And we had them doing bioaerosol sampling, CO2 monitoring, particulate matter monitoring indoors. Okay? And just to give you an idea, some of the things that they found, this is looking at the particulate matter concentration. So we kept them all in the hallway, and then they got to all go into the classroom all at once, as they would if you had a classroom in there, and you can see that the PM 2.5 concentration jumps up when they all go into the classroom and start kicking up the dust. So resuspension is real. Also, there was, if you look at the change in the increase in CO2 concentration, obviously when they go in, they're breathing out CO2, they increase the CO2 concentration, and this is looking at the delta here, how much it increased before they went into the classroom and after they went into the classroom. So I've proven that you kick up dust and you breathe, okay? But we also took a lot of swab samples, settled dust samples. We actually stashed Petri dishes, clean Petri dishes around the classroom and let them sit there for like 7 to 8 days. And they were still there 7 to 8 days later because we hid them well. However, in the middle of the sampling, we had some stashed outside. And these are, literally, they're an empty Petri dish. That's how sophisticated this is, but it does work. You get your settled dust sample. We went inside to collect the indoor samples. We went back outside and somebody had stolen 2 empty Petri dishes full of dust. And we're like baffled because there was all the equipment, but no, the Petri dish. That's what they wanted. So we found that really interesting. But this is looking at a schematic of here's the outside, here's the double doors that you have in this building, here's the entrance corridor, this is the outside, here's the entrance corridor, here's the hallway, and there's the actual classroom. And when we look at the indoor air of what we found in the indoor air and how it relates to all the bacteria, you see, for instance, that the outdoor air, this was a very well ventilated classroom, okay? We actually looked at the ventilation rate in the room. So the blue there is representing outdoor air. So actually it was a pretty well ventilated classroom and it kind of coincided with some of the measurements we had. However, when you look at the human associated, there was still a fair amount of human associated. And again, we didn't sample people. All we did was sample the doorknob as a surrogate for human contact because everybody touches the door on the way in. So we got this kind of composite human-associated bacteria, and it's also a dominant feature there. And then you see a little bit from the corridor, and all those other colors represent other samples that make sense in the context of what we did here. So this was a lot of fun. So I took a classic air pollution engineering class, and we looked at something different. So they looked at particles, CO2, but they also looked at bioaerosols, which, believe me, was very new for them. So I guess the takeaway that I want to give you here is that if we're looking at indoor microbiomes in the built environment, first of all, we are and have always been surrounded by a really diverse community of both bacteria and fungi. Okay? The sources and our potential exposure routes to these microorganisms vary. Okay? It can be affected by the water, so what kind of water source you have, whether you're on a surface water source, a groundwater source, whether it's a shower's on all the time or not, whether you're using low flow or high flow shower heads. That's a different projects that I've been doing with my colleague Maria King at Texas A&M. How well ventilated the space is. Are you getting a lot of fresh outdoor air in, or are you not? Are you just recycling indoor air? The type of building, how it's being operated. We've looked at homes that, kind of standalone homes to, very small, highly occupied manufactured homes, in the semi-rural. I also want to make the point that although there's a lot more press perhaps about potentially negative consequences of microbial exposures, there's now a whole line of research both within medicine and in other areas looking at potentially positive or protective microbial exposures. So that's an area that I'm very interested in because it'd be nice if I not only know what you shouldn't be exposed to, but perhaps how we might be able to design our buildings, design our water systems, etc., to actually promote beneficial exposures. Now that certainly involves working with people who do exposure work and certainly people in the medical field. Some other takeaways, I do like a good plate. Bacteria, there is very much a human-associated component of the bacterial communities indoors, but it's not everything. It's like 20%, maybe 30%. What's the other 70%, right? So we start to look at these other communities. Is it the ceiling biofilm in your bathroom? Is it something else, okay? Fungi are very much kind of an outdoor in. They're very much from place to place. They've shown there's a gradient of the fungal communities. It depends on where you are in the world to what fungi work their way into the indoor environment. With that said, once you introduce moisture into the indoor environment, then you're going to select for certain fungi. And if you've ever had a water leak of any appliances, which I unfortunately have, you will get to experience that firsthand. Or flooding, if we're talking Texas, right? Flooding. So I guess my takeaway here is, I know this is out of perhaps most of your fields at the moment, but I guess I'm arguing that engineers must be involved in this field. We're the ones who are supposed to be designing healthy buildings, the built environment, our infrastructure, water, buildings, ventilation. And in order to do that, we need to understand these interactions, and potentially positive ones and potentially negative ones, and also, kind of a plug for programs like the UT Biome and other things where you're involving the students, both undergraduate and graduate students in this work. We really need to train the next generation, and I could argue the current generation of engineers, to appreciate the microbial human health implications of how we design buildings and how we operate these buildings. And with that, I'd like to acknowledge a whole lot of people, most of which are here. Some are blurrier pictures than others. Many of these folks were involved in either mapping the UT Biome or in the HUD-funded project, and also our sponsors, HUD, NSF, the Sloan Foundation, the LIFT program for the UT Biome. With that, I'll answer any questions you guys might have.
Okay. One of the takeaways would be that the building should have more outdoor air coming in. I mean, basically, less sealed.
Yes, you should have fresh air. Yeah, and but there's a trade-off, right?
But we're going away from that. We're sealing.
I know.
5X.
You're going to make me say it?
Don't open and everything else.
Right. I would argue that you need to maintain adequate fresh air, and for a multitude of reasons. But one is the microbial, and Rich can talk about rebreathe fractions, which I don't think, well, the less fresh air, the more you're rebreathing the air of the person next to you. Okay, so you can kind of think about that a while. I would argue that yes, we need to maintain adequate fresh air so that you don't develop unhealthy indoor environments. Yes. And it does have energy consequences, right? So you have to be able to do it in a way that you don't have severe energy consequences.
That would only be valid during the non-cedar fever season.
I know exactly. I knew you were going there. And in fact, that is so true. In fact, I've got these dust samples, and if anybody knows a cedar pollen expert, one of my daughters is very allergic to cedar, and I have developed an appreciation for cedar pollen, and how do we design our ventilation systems so we can filter the pollen out yet let fresh air in? That maybe we just filter the outside air before it comes in, but we're still blowing in outside air and making up that mix of more fresh air versus recirculated air. So I don't say don't filter it, especially since I've seen the consequences of severe cedar fever.
We talked about molds, the fungi, and the bacteria, but another kind of microorganism is the virus.
Yes, aren't they fascinating?
Yeah, and so any
insight on that? Well, I've not done viral work directly. However, with that said, I teach the engineering microbiology course, and I thought viruses were so important that I asked actually Dr. Maynard from chemical engineering, who does a lot of viral work, to come in and give a guest lecture to my students about what are the viruses of concern, and how are viruses, for instance, different than bacteria and fungi. Certainly, there are known relative humidity and temperature effects on the survivability of viruses indoors. There are also, again, this rebreathe fraction during cold and flu season, for instance. If you're in an area with a very high occupant density, and this is sort of what you already knew, and you're not getting a lot of fresh air, and there's a lot of surface contact too, there's a lot of research going on how much of it is airborne and how much of it is the virus gets laid down by the previous occupant, and then you come in and haven't washed your hands before you eat. Whoops, sorry. So there's a lot of research going on actually at the CDC to try to understand what temperature, relative humidity conditions promote the persistence of viruses indoors, and also to understand how much of it really is airborne and how much of it is really contact transmission from person to person. So there is a very active area of research on that, and we have, for instance, gone back to even the filters that we had from the retail environment, and there are some double-stranded DNA viruses that we could then probe our samples for, and we found them. So we can detect viruses, for instance, in the filter dust. We did very few. And so we are looking to expand into the viral area because I think it's, as you know, hugely important. But we focused so far on bacteria and fungi. But it's a very active area of research, obviously, with the different viruses of concern that are developing.
So, Carrie, can I add something to that?
Yes, don't repeat your neighbor.
the #1 occupation for upper respiratory infections in the United States?
Is filter?
Anybody
No.
guess? Elementary school teachers.
Oh, yeah.
Highest incidence of upper respiratory
I
infections.
call them the cute contagions.
And so we've been working with school districts in Central Texas to try to convince them to increase their ventilation rates during the cold and flu. Viruses tend to not like higher relative humidity. They tend to thrive at lower relative humidity,
Yeah.
which is sort of
Counterintuitive.
contradictory with the
Yeah.
other biological stuff that we tend to deal with, which tends to like, especially mold, fungi, higher relative humidity. So there's this sort of trade-off there but I think in cold and flu season, that's one of the ways of ventilation and trying to keep relative humidity a little bit higher if you can in
And there's
classrooms.
other folks who are looking at cleaning practices for the stuff that they think is surface contact. You know, somebody with the flu or whatever touches something and then the next person comes along. And so there's actually folks who are looking at different cleaning practices to see which ones actually reduce the number of viral illnesses. And schools are certainly an active area of research because they're the cute contagions, but yes.
Has your data shown any comparison in the age of the house? You know, like I live in a 101-year-old house, okay, which is very drafty. As opposed to some of these newer homes that are very tight.
Yeah.
Especially in my buildings. This is a new building, so it's probably very tight.
We are actually so we call it the metadata file, which means it's huge. So we have these millions of sequences. For instance, the homes that we're looking at, were 60 homes visited twice, and they very much varied in how tight the homes were. So we measured how tight the homes were. We got estimates of that. And we are looking at the age of the home as one of the factors, the occupant density within the home, certainly whether there's children with or without asthma in the home, whether there's pets in the home. And we're also looking at, so we're trying to compare what we found, and I just showed you some of the early results, what we're finding in terms of these communities and how they tie to different aspects, you know, like the bacterial diversity. I mean, I think some of the original work, and again, we're still working this out, is higher occupant density seems to be leading toward higher bacterial diversity, but we need to confirm that with the entire data set. The other element is, for instance, in our other early work that we had done with Federico Norris, was a PhD student who kind of pioneered this filter forensics idea with Jeff Siegel, who's on our faculty, looked at lead. And age of the home, and guess what? Lead in the filters, those correlated. So we didn't see it as much for the other heavy metals, but it certainly correlated with lead. So there are certain contaminants that very much correlate with that and others that don't. But we're looking at that within the portable classrooms because their ventilation characteristics vary considerably, actually. And we're also looking in the permanent buildings or permanent classrooms. And we're still exploring just those questions because we want to understand, as you tighten up the home, you know, what happens to the bacterial diversity or the fungal diversity? Is it better? Is it worse? We don't know.
Yeah, but you also internet pollution
Huh?
You have internet pollutions too, you know, just for cooking,
I
and
know.
that's another story.
That is another story. Put an event. That's what I did when I finally renovated my kitchen after a decade. Is all I cared about it was event.
Outside.
Outside, somebody else's problem. No, I'm kidding.
I think we have time for one or two more questions.
Just real briefly, and this is not from an engineering point of view as much as it is from a mom point of view.
Yeah, how do you think I get into half of these
Yeah,
studies?
I don't believe in a 5-second rule where you drop it on a coin and still eat it. But I look at the dog, and the dog will lick the floor, will lick anything, will eat anything from outside and come in. And I know that if a human did that, we would get sick.
And what is so special about those dogs?
That's
Yes.
right. And so I'm saying it's not just an environmental air conditioning question.
It's also-
more than that.
Yeah. Because the human response versus the dog response. Well, you know they're doing the Human Gut Project. They're looking at me like we're nuts. Okay. They're doing the Human Gut Project. They've been doing it for a while. Thousands and thousands of Americans. Have sampled the human gut to see what does the human microbiome look in the human gut. And they've done that across the United States, and then they extended it to look at other countries. And guess what? We have a Western gut that looks different than a developing country gut because they have a very different diet. So there's also the diet impacts on all of this for things like the gut microbiome. But you're right. But actually, in defense of dogs, what they're leaning toward now is they think they're beneficial with respect to asthma. I like my dog either way.
First year.
What? Huh?
First
Yeah,
year.
first year. That's right. And these things, one of the things you have to keep in mind is when you read these things, they're absolutely right. You know, so when I said that what was found to be protective, the farm-like microbiome, that's only if the kid from 0 to 1 years actually had active exposure to that environment. Not that they lived on a farm from 3 to 5, etc. So it's a very specific timing of the exposure too that's protected. So I don't know if everybody wants to run out and visit a barn, but I think it's too late for us now.
What about a comparison with dog to cat? I would think cat would be a lot different.
I think a cat is very different, and I just heard from JP, who's in Finland, and was at Chicago before that, talking to a bunch of folks who do the microbiome of built environments and different things, that there are some folks looking at the cat microbiome too. So I guess there's a study, he mentioned that they're about to start looking at the cat microbiome. So I think we'll get a better understanding. I think in the particular study there were more dogs than cats that happened to be present. And so they didn't get a statistically representative sample of what the cat had done to what cats might do to the indoor microbiome. I think it was Dunn who had done that study. So I'm sure somebody's looking at it because there are cat lovers and there are dog lovers.
But there has been great studies on cats as samplers for the indoor environment because cats, walk all over the floor and then they tend to lick their paws.
So they ingest it all the time.
Some people have donated their cats to science, and if you look at the accumulation in the fat of cats, it's a great sort of sentinel
biomarker
or marker of the chemicals that we're exposed to in our everyday life, mostly because they're licking all the chemicals off their paws. So pets can be very valuable sources of information if we
And I've
use
had,
them right.
on the record, I've had cats and dogs, so I'm both. It's all good.
I think we need to wrap up now, so I want to thank Carrie for...
All right, I hope you enjoyed it.